Thank you. I think you guys are hearing me, right? So Dr. Walker gave me a challenge today to do this in 20 minutes and definitely under 30 minutes. I'm going to try. I'm a little bit different in, yes, I am a bit of a hurricane. So I'm going to try to go through this presentation being a hurricane as well. So I tend to do a lot on my plate, especially when challenged. I am the CEO of a company. Sorry, 1 second. Okay. So I am the CEO of a company. Those are my disclaimers. Ventura Clinical Trials. We are a CRO. We have put out a lot of products into market for pharmaceutical companies. When the product became fecal material in a capsule, one could say I went rogue as a clinical trial investigator and wanted to understand why we are putting stools in a capsule and what is the whole thing about the microbiome, especially as I am a gastroenterologist that does fecal transplant. And one of my patients said to me, you know, you guys are putting mud on the wall and you expect it to stick. And I absolutely agree with that. We need to understand how the mud sticks on the wall. And we need to understand what is changing in the microbiome. What are we doing? I'm not hired by any pharmaceutical company to speak. I'm not paid by anyone. In fact, it's all my funding that funds my research. Albert Einstein, and I'm gonna say this, said, it is all research. If we knew what we were doing, it would not be called research. And I say this because a lot of parents ask me when especially with the whole Dr. Adams study that brought in a lot of kids with autism in my office, parents wanted a report. They wanted to know, what am I seeing? And the reality is this is research. We're all doing research and we need to understand that. A couple of acknowledgements. Dr. Stolman, 25 years ago, I was a fellow in GI, University of Florida, and I was presenting my poster on a clean esophagus, visceral hyperalgesia. And he said, Sabine, don't waste your time with the clean esophagus. The future is in SHIT and the microbiome. So essentially, here we are, 25 years ago, he was right. Second acknowledgement, Dr. Tom Barodi, who has been a mentor to all of us gastroenterologists that have done fecal transplant, has certainly held the hands of everybody as he is the pioneer, the cowboy that started with the vision of using stools and changing the path of a disease and not just C. diff. The third acknowledgement is Dr. Sidney Feingold. I think it's very important in our path of discovery to acknowledge the people that have got us to this level, right? And Dr. Feingold was the physician who at 97 years old passed away, wrote the book on anaerobic bacteria. And most of you know him because he was the one that actually started the whole vision and the whole opening our eyes to the fact that autism was a gut bacteria. So I went to Dr. Feingold when I discovered myself as a gastroenterologist that after doing fecal transplant on a patient that had Alzheimer's, the patient 6 months later came back and remembered his daughter's birthday. It shocked me to the point that I said, well, what did I do? What did I change in the microbiome that had this guy remember the daughter's birthday? So I went to Dr. Feingold and he said to me, here's a paper. I've cultured the bacteria behind Alzheimer's for the last 40 years. And he had Alzheimer's himself. So he wanted to understand and crack the code. And he said, when you get your own genetic sequencing lab, you're going to understand the microbiome and you're going to understand and prove my paper. And so he passed away and here we are. I'm speaking here on autism, almost divine intervention in a way, as I feel that, he's passed on all his work, etc. I'm going to start with, yes, 2,500 years ago, Hippocrates was ridiculed when he said, all disease begins in the gut. But it's not only Hippocrates, Maimonides, and I know Dr. Gershon and Dr. Krigsman know Maimonides very well. He was a rabbi and a physician. And he said, no disease that can be treated by diet should be treated by any other means. 1849, William Osler, the father of medicine, said one of the first duties of the physician is to educate the masses not to take medicine. 1861 to 1931, the aim of medicine is to prevent disease, prolong life. The ideal medicine is to eliminate the need for the physician. Where have we gone wrong? 4 billion prescriptions in 2011. And believe me, I'm part of the problem. Yes, 300 clinical trials between me and my sisters, Dr. Adams. We've done a lot of clinical trials for pharmaceutical companies, but we believed that if a drug was a good drug, it should go to market. And if a drug was not a good drug and had side effects, it should stay. And so we've stayed in the trenches as investigators, working with the FDA because of that simple principle that good data shines, bad data should stay back. And so we have been part of the problem, but unfortunately, like medicine and science, we only know as things progress. And here we are with autism. And autism is really a vision on the microbiome. Because if you look at 1980, 1 in 2,000 kids had autism. And now it's close to 1 in 54 and 1 in 40. But it's not only autism, it's actually obesity. We used to have a problem with malnourishment, and now we have a problem with obesity around the globe. In fact, islands in Kiribati, which are little islands that used to survive on fish and vegetables, are now morbidly obese. And that is a problem. And it's not only obesity, it is COVID. What is going on with COVID-19 around the world? Is our microbiome so altered? And do we have such an imperfect microbiome that we received from our great-great-grandparents that is now so altered that disease and viruses are penetrating? So unfortunately, we have become drug America, and it's not only drugs of the pharmaceutical companies, it's also the herbs, it's also the vitamins, it's also the supplements, it's also the foods. One thing America is big on, it's consumerism, marketing, branding, the whole idea of branding. And so we tend as Americans and as America to give the example to the rest of the world about what we should eat. And everybody starts thinking that they need the same pill that everybody's using to help their kid. Marketing 101, we are being sold. We're being sold mega consumptions of food, beef with antibiotics, supersizing everything. And unfortunately, that creates a globalization around the world. And with globalization comes globesity. But unfortunately, it's not only obesity, it is autism. It is celiac sprue in India. The fact that China has ulcerative colitis, that is a problem. If you look around the world in India now, 1 in 100 kids in India is autistic. And in 2017, this is the map of the world. It's looking as bad as COVID, frankly, because there is something that is changing in the microbiome, in our microbiome, that is causing all these diseases. Now, if you look at the data of autism, and I was kind of Googling the Google because autism is not my forte. It came from Dr. Feingold and Dr. Barodi and all this incredible work from Dr. Adams. If you look at the etiology of autism, there's a thought that it was genetics. But when you talk to patients, and as patients have come into my office, I take a thorough history, and very few of them actually have a genetic predisposition or have family members that have autism. Of course, another hypothesis is neurodevelopmental, and then, of course, the environment. Which brings us to Dr. Feingold, and how did I get, and I'm re-looking at my timer because I gotta be on time. Dr. Feingold essentially had a beautiful collection in his freezer at the VA of autism, where he started collecting stools. For those of you who don't know Dr. Feingold, published 392 papers, 118 review articles, 39 books, 201 book chapters. He postulated that autism was caused by a bacteria, the Vibrio, and Clostridium perfringens. In 2018, September, he passed away from Alzheimer's. And on November 8, 2018, he passed on all his books, as I was during the Woolsey fire. If a microbe can cause autism, then perhaps microbiome fecal transplant can change and rebalance, right? But when we think about autism and rebalancing, I think it's important because everybody looks at that, and I've seen a lot of home therapy and people giving advice on others on how to do home fecal transplant. You've got to make sure that one, you have the right microbiome that you're transplanting. And I've been doing fecal transplant for too many years to know that when I do it for fecal transplant for C. diff, it is important to have the right microbiome, to do the right procedure, to remove the dangerous cells potentially that could be planted in there, to know who is your donor, because you do not want to swap, you know, fecal transplant of autism for something else, right? So I think the physician from Italy said, you know, it's like fecal transplant is like driving a Ferrari. I have to say it's actually like driving a tractor. And so you have to know how to drive the tractor, and you have to understand. So we're, you know, parents with autism. And as I started looking into this world, I realized there's a lot of stuff out there. You guys are being sold, you know, probiotics, this and foods and this nutritional compound, and you're all looking for answers. And I'm here to kind of help out also with the truth, because we need to understand. And an old professor of mine used to say, it's all about show me the data. So I'm a big Trek Star Wars girl. Captain Kirk used to say, the greatest danger facing ourselves is an irrational fear of the unknown, but there's no such thing as the unknown, only things temporarily hidden, temporarily not understood. Really, he was speaking about the microbiome. So what is the microbiome? It's a collection of genomes of microbes in an ecosystem. I like to bring it back to the transmission of a car. So when you look at that big log in the toilet, and sorry to be so graphic, but this is, you know, what we're talking about, it's like a transmission of a car. When you break down a transmission of a car, it has 880 pieces. Now, at any point, any piece of the transmission could be broken. And unfortunately, you could fix that piece, but then you're going to break another piece and another piece. And eventually, you're going to be needing to change your transmission. Now, you're going to use the transmission of a Honda into a Mercedes, or a Honda into a Ferrari, or a tractor? No. So that's why you have to be very careful when you do fecal transplant, and selection of the donor is the criteria. No microbe is an island, because it's the same thing in the microbiome system. You cannot just give penicillin to kill a streptococcus and then expect it not to do any damages. And back in the days when we didn't know and we just gave penicillin, we didn't know we had other bacteria that we had to worry about, like the bifidobacters and the lactobacillus. And so, you know, science progresses. We start understanding what we're doing with one more research after another. So killing one microbe affects many. Killing one creates a dysbiosis. I like to think of it as an imbalance, the good versus the bad. I like to also think of my friendly bacteria and my unfriendly, which are the pathogenic versus the non-pathogenic. What is interesting in the microbiome is that the pathogenic or the non-pathogenic can become pathogenic at some point. So when you have this disruption, this dysbiosis, of course, bacteria penetrates, goes under the colonic membrane, kills off your immunity, and there's the cascade that starts of autoimmune process. Like Dr. Adams, weeding is actually my example that I always give to patients when I do fecal transplant. I say it's like weeds in a garden, and you have to remove all the weeds. So I always bring back to gardening, because gardening is my favorite thing to do. If you look at a beautiful forest and a beautiful garden with flowers and trees, etc., and you think of that as your beautiful microbiome that's at peace, and all of a sudden you cut out a bunch of trees, then the birds don't have any place to go. The bees don't have any place to go. Pretty soon you have a desolate planet. And it's the same thing in your gut. If you kill off the Clostridiums and you kill off your Bifidobacter and your Lactobacillus, you're left with a bacteria, Clostridium difficile, that starts flourishing and takes over and then secretes its toxin to kill you. And that's how I like people to think about it. But I also like to think about it as humanity. Humanity is beautiful because we're all different. We all add a role. So if you look at humanity as a whole and kind of compare it to the microbiome, and you remove a race, then all of a sudden you have a bunch of robots. Well, that's not really a healthy planet, and that's not a healthy humanity. Do we not risk killing humanity as we go on? So diversity equals health. Loss of diversity is disease. How do I know that? 42 clinical trials. Now, people have said to me, why are you doing 42 clinical trials at the same time? Shouldn't you just focus on Alzheimer's or autism or Crohn's? The problem is you cannot. In the world of microbiome, you have to look at all the diseases to understand what is going on in the microbiome space. So you need to understand what's going on in Alzheimer's, in Crohn's, and maybe find correlations that are there. The one thing we did notice, however, is that we are all different. Now, I didn't need to do, an expensive study of 26 patients to show that we are all different. We all have different fingerprints. It obviously makes sense. We all have different microbiome. What is interesting about the microbiome is that if we are all different and one of us has a lot more bifidobacter and is healthy and another one has a lot less bifidobacter and is healthy, how can we compare those two people, right? So when you go to these labs that you're all sending reports to, and they send you a report that compares you to a healthy person, let me tell you, there is no such thing as a healthy gut microbiome, because we all have different things, different disease, we all take different probiotics, we all take different foods, we all eat vegetarian, vegan, kosher. All that impacts the microbiome. So for a lab to show accurate data, they must be CAP and CLIA certified. I'm very big on the regulatory overlooking things. Show a valid, verified, reproducible assay. In other words, if you give your stools to someone, you should be able to reproduce that report in 3 months, 4 months. There's not that much variability in the individual when you look at the microbiome, and preferably the assay needs to pass FDA clearance, because we are all different and there's no other way to compare. But we can compare, and what I have done is compare the family. So what we see here is actually my family portrait. And again, my timer just went off. My family portrait, and what you see is my husband, me, one of my daughter, and daughter number 2. Now, I wanted to look at my family because I have one kid that eats a ton of food and is super skinny. And I wanted to know, well, what is different in hers? So what I noticed is actually there is a difference in her microbiome, and I know which bacteria and group and the metabolomes of this bacteria. However, I am not going to go rush and put that bacteria in a yogurt, because I may create an imbalance in my own microbiome. And therefore, this is why we shouldn't be rushing with all this till we understand the whole microbiome, because I do not want to change my microbiome where I have extremely thick hair to be skinny, to a microbiome that is potentially making me lose hair. So this is why it's important. Now, the other thing that's important when we look at the microbiome is that it's very important how we look at the microbiome. So this is a pipeline that I use, and that the company that I do, the bioinformatic pipeline that I use, and I think the FDA uses this pipeline as well. When we look at the microbiome, it's important to look at the surface and then go all the way deep into the species. So when people say, well, what is in my microbiome? It's not as important as how much relative abundance, as much as, well, what are your Lachnospiraceae doing? What are your ruminococcus doing? What are your bifidobacter? Because everything is in relationship. You have to analyze the microbiome within the relationship. It's very important to also be humble and to realize that in the microbiome space, and especially as we enter a world of fecal transplant, we only know the 1%. 2% is the stuff we don't know. And then there's the 97% is the microbiome that we don't even know we don't know. So I think it's important when people look into the microbiome space to realize that there is a whole world out there of microbes that don't even have a name. So Clostridium perfringens sure could be one, but there's something else. So it's important to look at the families, but it's also important to look at the individuals and compare it. What did we realize? C. diff is in everybody, and actually C. diff is the model of dysbiosis in humans. We started this whole mission because of C. diff showing that there was a improvement with fecal transplant. And I'm going to skip fast because I'm approaching my 20-minute mark, so I don't think I'm going to make my bet. C. diff obviously causes colitis, the treatment flagyl, vancomycin, fidaxomicin, clinical trials. When clinical trials don't work, probiotics, and then fecal transplant. Fecal transplant, as you know, is the process of changing stools to another patient. Started in the 4th century, was thought of by Dr. Einsman. When we do fecal transplant in the sigmoid for C. diff, we achieve 82% success. When we go all the way to the cecum, we achieve 92 to 99% success, but that's only in C. diff. C. diff, we found with our assay that it was an imprint of everyone. However, when we changed the pipeline and we looked at the metabolomes, we discovered that the ones that actually had C. diff toxin fit a criteria. So this is when I do fecal transplant, I like to look at my patient and I like to look at my donor. And I like to follow my donor to see whether the engraftment matched. This is a paper that we did that was acknowledged as best poster at American College of Gastroenterology that basically showed that if 2 out of the 3 donors had a matched donor and succeeded, and the one that didn't was a reject, was kind of on the outside. When we use the Bray-Curtis distance matrix, we also see that the 2 successful are in the middle and the one that rejected are at the end. So it's important. So fecal transplant obviously opened up our eyes because this is C. diff before and after. Dr. Barodi talked about the capsule with Crohn's disease, and we have seen that with Crohn's disease. This is fecal transplant before and after alopecia areata. Cases of acne, obviously Dr. Adams' cases with autism. Dr. Jessica Allegretti, her mice study showing, obesity can be transferred. But so all these are great and great anecdotal cases. However, there is still a potential, the same mechanism that grows hair, changes Crohn's disease, cures, improves cancer in some patients with Dr. Wang, Mimi Wang from MD Anderson, can also cause rheumatoid arthritis, can cause Sjogren's syndrome, obesity, and sepsis and death, as we have seen. It is important to understand the microbiome before we start thinking about doing fecal transplant. This is a case of mine where the mother has a lot of diversity. The kid had almost an overgrowth of a bacteria and no diversity. We altered the microbiome. I don't like to call it fecal transplant anymore. I call it refloralization, because refloralization, bringing back the flora from the flora. And the patient, after that, improved his Crohn's disease and is actually off Remicade and doing amazing, showing the data of Dr. Barodi. This is another case where mother-child, the child is here, and we basically improved his fecal transplant and his diversity, and he's resolved with Crohn's disease. Now, when we look at autism, and this is why we're here, is basically, I like to look at the families, and essentially when you look at the families, you start seeing that this is a mother. This child has an overgrowth of a certain bacteria compared to this other child. And you could see the Shannon index is way lower, agreeing with actually what Dr. Adams and Dr. Rosie found. Same thing in this case where it's a mother, child 1, child 2, child 3. So ideally you could kind of almost say, well, I could use this microbiome and transplant it in this child, because these 2 kids have a lot of diversity and seem to be fine. But this is what needs to be done in the future to look better into fecal transplant. Same thing with this. We see mother, child, overgrowth of a certain bacteria. Again, same thing. So all these cases kind of could be succeeding and be in that 50% group that Dr. Adams was talking about. But then you get into these cases where you look at the microbiome and you see that the Shannon score index is normal. They have a lot of bifidobacter, a lot of Clostridium. And you ask yourself, well, would that succeed in fecal transplant? Second case, same thing. Would that succeed in fecal transplant when you see a microbiome that is diverse, that is improved? The problem is a lot of parents are doing so many things with these kids and giving them, so many products. It's very hard to see the microbiome at the way it should be because you guys are putting a lot of band-aids on it. So we cannot figure out whether this is, because I think what's going to happen in the future is we're going to figure out that autism is either a primary gut problem or a primary neurological problem. And you've heard it here. So Dr. Krigsman, Dr. Adams, Dr. Walker, and everyone, this is what I believe. We have to start thinking that maybe there is a gut entity and maybe there is a brain primary entity. As far as Prevotella, yes, we did see a lot of patients with Prevotella. However, we also did see in this family a kid with autism that had a lot of Prevotella compared to his family and his sister. Clostridium perfringens, we found it in a couple kids. We did not find it in a lot of other kids. Of course, we're the beginning of this. I always tell parents we're at mile 1 of 300,000 of trying to figure out the microbiome. And trust me, I'm just a hurricane to speed it up. Desulfa vibrio, we found it in one pretty relatively abundant compared to mother and another child. However, super high in another kid. Bifidobacter, we found it super low in one kid compared to his family. And then we found it super high in one kid compared to his family. So all the possibilities are there. This is why we need to do a clinical trial on the microbiome and autism. We started this in 2019, September, or 2018, or 2019, sorry. Time is just passing by with COVID. And we have now also been approved to do fecal we were approved to do familial fecal transplant in autism by the FDA prior to COVID. COVID put a stop on everything with fecal transplant. We are actually developing an assay that we're working with Dr. Alex Kurotz and other labs to show that there is no COVID-19 in the stools. We're actually the only lab that picked up COVID-19 in the stools as whole genome sequencing in patients that were positive throat swabs. So we hope to use this assay to advance the world of fecal transplant. We are also going to be planning to help Dr. Adams with his study because we are a CRO, we are a research center. This is what we do for a living and we want to try to help kids. I'm going to skip that. So essentially all disease begins in the gut. I invite everyone to the Malibu Microbiome meeting March, where all the fecal transplant doctors have assembled as a biome squad, including Dr. James Adams, as he's part of the group, to try to understand these microbes and to try to understand more of the microbiome world as we get into the fecal transplant route. Especially we have to go faster before the products are coming out. And believe me, in my desk as a CRO, I've had pill poop from Korea, China, Japan. So I see the future. The future is going to be fecal material in a capsule, and that's a scary future for me. And so we have to figure this out before we step into that future. I'd like to thank my Progenabiome team that has followed me in my path of being a hurricane and have caught up with me. And then of course my advisory board, and that's about it. So I'm open to questions. And I think, hold on, I will see 28 minutes. Dr. Walker.
All right, thank you very much, Sabine. Time for a quick question from the audience, if anyone has one.
There's one. Do you use your own proprietary assay for stool genomics, or do you use a commercial assay?
No, I use my own. We have SOPs. We are a lab, genetic sequencing lab, Progenabiome. Actually, we took over for what Ubiome tried to do, except we're doing it the right way. We're not using, mice poop as our controls. We're looking at the microbiome with diseases, with patients, with families, and yes. So it's our own assay.